I have third lecture, and today I'm going to focus mostly on the metabolism and related activities. Ilamas, you can start recording.
Yes, sir, I started.
Okay. So before going into the lecture, I just wanted to tell you about the TA, and tomorrow you will have your first tutorial. And if you look into that, you have been already assigned a room on the team. If you have not seen your name in the team, then do let me know. And for the D1 batch or D1 division, we have rooms from 11 to 21, and each room has around 35 students. And for each room, there are 2 TAs. So this is the list of all TAs. Now, and tomorrow they will basically revise few concepts and tell you about last week's lecture. Okay. So now moving on for today's class. So these are the objectives for today's class. First, we'll talk about. I'll discuss about the membrane protein and its function. Then I will come to the metabolism, the concept of energy currency and standard and cellular conditions, and what is the exactly currency of the metabolism, that is ATP. So first of all, I'm going to start with the membrane protein, and these are basically very important class of proteins in the cell. What you see over here is this is a lipid membrane. And this is, basically the lipid membrane over here. What you see is there is a head group and there is a tail group. So the tail group over here, that is basically hydrophobic, and the head group is hydrophilic. And this is how the membrane, basically this membrane, protects the cell, and it make it intact. Otherwise, you can have a lot of things getting inside the cell. And but then, in order to have that regulation, which things has to be brought in, which things has to be brought out, there are micronutrients, so there has to be kind of sieve which are selective, which should be selective, and apart from that, there has to be some kind of other functions like cell-to-cell recognition, all those things. For that kind of activities, for that kind of purposes, the membrane proteins are extremely important. Over here, you see a different classes of the membrane proteins. Basically, this is basically, you can see this could be a signaling protein, this could be integral protein, and we'll talk about that in all these kinds of membrane proteins. But apart from that, what you see is just across the or on both sides of the cell or this both sides of the membrane, there are some structures. Specifically, it could be fiber, extracellular, or this could be cytoskeleton. So over here, what you see is microfilament, and that is again somehow in connection with this membrane protein. So it is a very dynamic it's a very complicated system. So here, I'm just showing you one example of membrane protein. This is ryanodine receptor. The short form is RYR, and it's basically meant for the intake or the intake of calcium ions inside the muscle. So what happens is the calciums are basically pumped from this side to the side, basically inside, and then there is a selective mechanism for it. So what you see over here is this orange part, that is mostly that is very selective, and it is selective for only calcium. It doesn't allow any other ions, but because of the structure, because of the architecture, these particular proteins are extremely sensitive towards particular ions, particular nutrient, you can say. So on the right side, what you see is the opening and closing movie of this particular channel, and now you can see it's opening and then it is closing. So all this structure, what you see here, that is made of it's a long amino acid chain or polypeptide chain, you can say. And over here, there are different subunits. Again, I'm not going to get into all that, but then I want you to appreciate how complicated this system is, and yet it is very selective and very specific, very dynamic. So these are the different types of the membrane proteins and their particular functions. The first type of the protein is called as transport protein, and this particular transport protein can be very selective towards intake of few nutrients, and also with the help of ATP hydrolysis also it can take out some nutrients or take out unwanted material from cell to the extracellular site. Then the second is the enzymatic activity. This membrane protein at times can perform the enzymatic activity. And specifically over here, what you see, it's a classic model for the enzymatic activity. You can say lock and basically what happens is particular ligand, or this ligand could be anything. It could be anything inside the cell, which means it could be protein, it could be some micronutrients, or it could be again other biomolecules. So what happens is it can bind to this membrane protein, and it can catalyze a few things. So basically there is an active site over here, and with that active site the substance is getting into this particular ligand can basically get activated, and then there could be different types of products can be obtained. Again, this is very important in terms of the metabolic pathways. The third type is the signal transaction, and signal transaction is again very dynamic process. So in this particular, what happens is in this particular class of protein, the protein will bind, protein binding site has a specific shape, and with that specific shape, the chemical messenger over here, what we see in the orange, that can bind, and then after binding there could be some structural changes of this protein, and that due to that structural changes, signal can be generated, and that signal can be transduced. So typically this could be, this chemical messenger could be some hormone, and secretion of hormones might activate few pathways. Again, it's again part of the metabolic process itself. And that is why before talking about metabolism, I wanted to introduce this topic of the membrane proteins, because you might be hearing about all this membrane proteins in the subsequent lectures. And then we have the fourth class, that is called as cell recognition. And in this particular class of the membrane protein, basically what we see over here is there are some glycoproteins which are, you can say, post-translation, after post-translation, and then this glycoprotein can identify few tags, and basically it can few tags by recognizing the membrane proteins of the other cell over here. So basically this is one cell, and this may be other kind of cell. So because of there is interaction between the glycoprotein and the other protein so because of this particular interaction, there could be recognition between different kind of cells, and that's also performed by this membrane protein. Then you have the fifth class called as intracellular joining. So in this particular class of protein, the adjacent cell may hook together in various kind of junctions. So over here you can see some kind of curvature over here, and basically there could be some gaps also, and there could be some kind of curvature, and because of that there is a binding between these two proteins, and because of that binding you can say that there is a intracellular joining. So these are very even though the forces over here involved, it could have some electrostatic interaction, it could have some van der Waals interaction also. Then the last class is about the proteins which can attach the membrane protein which can attach the cytoskeleton and extracellular matrix. So over here, what you see, this is the cytoskeleton, and this is extracellular matrix. And this class of protein can interact with this fibers within the cell and outside the cell. So and then with that interaction, it can maintain the shape of the cell, and it basically stabilizes the local. So basically, it can stabilize the localization of few structures, which are very important for the cell. So these are basically different types of the membrane protein, and now I will move to the metabolism part. And first, basically, we have to understand why cell will need the metabolism. Why? I mean, what is this metabolism, and why it's needed inside the cell? So here, to give you analogy, I'm just going to start with this map. And in this map, what you see is there are different routes between cities inside the India. And you can see, it's extremely dense, and it could, you can see some red dots, you can see some black dots, and then there could be very, heavy network in between this red dots, and then, the traffic between the other black dots is not that heavy. So in particular over here in this, basically what you want to know is this map does not show how many flights are in between this particular route, and so basically when one has to take flight from, let's say, Mumbai to Delhi, does one have to travel to, let's say, Nagpur, and then from Nagpur to Delhi? So who decides this? Or one can travel from direct Mumbai to Delhi. So what are basically different needs to have the different routes? So again over here, this is the different map, and it shows the map from the Rome. And over here you can see a lot of dots. Obviously this map doesn't exist. I mean, the Rome doesn't exist at this moment. But just give you analogy. And over here, what you see, there are some specific landmarks. And so again, what we see is there could be many things over here, and we have to understand what these things could be. So just like this, we have chemical inside the cells, and you can see this is some kind of picture. And within this picture or within this map, you can see many chemicals. Now, how do I connect them? And so basically, do I have any idea how to connect them? And this particularly, I mean, how this basically one chemical is related to the other, that can be found by this metabolic pathway. And over here, there could be, I mean, we are not going to talk about this metabolic pathway in detail, but then during respiration, during the photosynthesis process, there is a breakdown of few chemicals, and there is a order in which that breakdown happens. And because of that, there is some relation between relation between these compounds or chemicals. And that is basically, that determines the metabolic pathway. So basically, over here, in the few first first two chart or map, what you see over here, the dots are basically your chemicals, different chemicals. And the in this, in this context, basically what you see, the routes, those routes are nothing but the metabolic pathways. And so basically, for one instance, the metabolic pathway might take the route from Mumbai to Nagpur and Nagpur to Delhi, but the other route could be, in the most prominent would be Mumbai to Delhi. But for some instance, it could basically there could be some diversion also. So it's an extremely complicated process, and today we are just going to discuss few insight about it. I mean, we are not going to get into a lot of details. So what I wanted to say that the metabolic pathway chart is just like airline map, and each dot represents a metabolite or a chemical, and can represent airport, and the line connects the compounds that can be interconverted or the airports that are connected to each other. So it's just the analogy how this metabolic pathway chart could be. But when you talk about the metabolism, it's basically totality of organism's chemical reaction, which means whatever is happening inside the cell, whatever happening inside the organism, there are numerous chemical reactions are happening, and during each reaction, there could be, the reaction can get into some different products, and so basically at each step you can see there could be many intermediate compounds also, or intermediate products. So as such, what we can say is metabolism is an emergent property that arises from the orderly arrangement or interaction or of a set of components or parts. And this parts or components by themselves do not have the property. So again, if you remember, in last lecture we talked about the self-assembly of the cells also, and this basically this metabolism, again, it's a very complicated process, and the way cell organize itself, it's very difficult, and how it survives, how it multiplies, how it reproduces, it's a part of the metabolic process itself. So there are many things over here. Now again, in first class we talked about few characteristics of the life, and two prominent characteristics were reproduction and growth and development. So when we think about reproduction and the growth and development, so these are basically reproduction. One can think as fundamental driving force for the organism, and I mean, there is no clear reason why it could be fundamental driving force, but because when you talk about evolution, you just imagine that it starts with the all the elements. Elements could be carbon, hydrogen, nitrogen, oxygen. These are the basic few elements, and apart from that we have other, it could be other elements also, but then evidently we see carbon, nitrogen, oxygen, hydrogen, and with this element itself, one can see that there is a rock which doesn't have any life, but these elements come together and they make a life. And in order to propagate that life, it constantly goes into the reproduction, and that is why I want to make point that since it's a reproduction, it's a fundamental driving force. So here, there are some few examples over here, and under these are different organisms, and under different conditions, how do they basically propagate, how do they make the reproduction. So it's basically inside when you have food scarcity, then there will be different different shape, and when you have the favorable condition, then it can take a different shape. So it is a very dynamic process, and it is regulated or propagated by genes which are inside the cell. Now, but then how do you basically this reproduction happens? There are there are biomolecules, and these biomolecules are DNA, protein, carbohydrates, lipids, cholesterol. Then you have the AMP, ATP, and so on. There are many chemicals over here. So, but they, all these biomolecules are there. But the biggest question is how does any organism get this biomolecule? And what are basically, again, when is it synthesized inside the cell, or how exactly it is generated? So it's it's a very interesting point to know. And when we look into the nutrients, now just let's take very simple organism, which is bacteria. And when you think about the nutrient requirement of the bacteria, there are in terms of the requirement, there are two most important requirements. One is source of carbon, other is source of energy. Now, in terms of the source of energy, it could be sunlight, and source of carbon could be atmospheric carbon dioxide. So basically, these are, when you think about the atmosphere, think about the earth, and this is what you want to think. But when, in terms of the bacteria, in terms of the organism, that source of energy could be glucose, and also the glucose can be a source of carbon. So apart from this, there could be, so the glucose in this case can be both source of energy and source of carbon. But apart from that, just like glucose, there could be other molecules which could be generating energy, and which could replenish the cell. And depending upon how the glucose breaks down, then the pathway and the corresponding pathway can be determined. So abundantly, when we talk about metabolism, commonly what we see is, most of the textbooks, they will talk about the breaking of the glucose, and from the glucose how one derives the energy. But this is major source of the energy, and that's why most of the textbooks talk about it. But apart from glucose, there are different molecules which can be still categorized as the source of energy or source of carbon, and we are not going to discuss that. So here what you see is typical, you can say, organism also. It's called as neurospora. And when you give very minimal medium, so the minimal medium could be just consisting of some few salts, glucose and vitamin, and you grow that on the some kind of support medium, that could be agar. Again, this the agar, biotin, glucose, inorganic salt, and also this all terminology seems very biological to you, but again, when you remember these things, I think glucose you might be able to remember, inorganic salts that you might be able to remember. Biotin, when you think about biotin, you think that as a vitamin. And agar is kind of a jelly-like substance, which is necessary inside the lab to grow this medium, or grow this plants, or grow this organisms. So in this, even though, let's say, you want to grow this particular neurospora, it can grow in the minimal condition, Also, if you want to basically have a complete growth or kind of wholesome growth, then you need to have this minimal media with different kind of compounds, which could be amino acids and few other nutrients, and those are basically provided in the agar. So with that minimum media, still it can grow. But it's a adaptation. You can say, to just give you an analogy, most of you might be knowing about this money plant, and money plant basically it can just survive on the it can grow just with the water, and with the water it is able to you know technically it injects all the water, and within the atmosphere, with the atmosphere it can take the carbon dioxide, it can synthesize the carbon, and it can basically synthesize the energy needs. Along with that, it can biosynthesize other compounds. So with just few basic nutrients, it can survive, money plant, and so it's a very extreme condition, and it is happily survive over there. So but then when we think about this metabolic activity, when you think about this salt, the glucose, the biotin, these are minimal media, and again over here the process could be, you have the energy source, and in order to biosynthesize particular molecule or particular thing, what would happen is the carbon source, which is nothing but glucose, that could get broken down, and after breaking down there could be two processes. It could act as a precursor for biosynthesis for the further molecule, or it can generate the energy, and with these two outcomes, again one can generate the biosynthesis molecule. One can have the biosynthesis of the biomolecule. So when you think about the metabolism as a whole, it manages the energy and material resource of the cell, and in particular the breaking down of energy, or breaking down of source, breaking down of your chemical, that is termed as the catabolism. And and then during that catabolic process, one can generate the energy, as well as you can have some precursors to the precursors for the extracting the energy. While in contrast to catabolism, the other process is called as anabolism, and over here in this particular anabolism phenomena, one can use the energy and precursor to build up the required biomolecule. That is nothing but biosynthesis. Now, when you think about cell, and when you think about the metabolism, and you think that all these carbons is breaking down, and basically there are different processes going on. So simplistic, when you think about this cell, you can think that as a thermodynamic open system, and where in this cell, when you think about the cell, and cell can exchange both heat and the matter with the surrounding, and with that it can do the work which is needed for the various processes, and also it requires energy, and with that energy it can uptake it can uptake some nutrients, then it can metabolize it and create the waste product, and whatever the waste product is created, that can be excreted from the cell. Most importantly, you have to understand over here, all the living beings, the living systems work within some framework of the physics, or some framework of the thermodynamics laws. So one has to basically consider all this, as a cell. When you think about cell, there it is governed by some scientific laws. It does not, things inside the cell just not happening randomly. There is a definitive science behind it, some definitive physics behind it. So when you think about conversion of energy, so you eat food, and from food you get the energy. So that is basically nothing but first law of thermodynamics. So here what you see is it is a bear, and it is eating the fish, and fish has this chemical energy. After it consumes the fish, and that chemical energy is getting converted into whatever work that bear is doing. And apart from that, there are some metabolic activities, and during that metabolic activities inside the bear, that that can create the biomolecules, biosynthesize them also. So basically, overall, what you see is here there is basically conversion of energy from one form to other form. Then here we can basically breathing, having some activity. Over there, so basically in this picture, what you see, this bear is running, and now with this running, it is, again, many things are happening inside the body, and it is basically radiating some heat, also breathing out carbon dioxide, while having the metabolic activities, it is, producing some water also. So overall, the entropy is increasing because many things are over there. It's just all the many things inside the beer, many compounds, they are breaking down, and this breaking down will bring the disorder, and overall this will again contribute to disorderedness to the universe. And that is exactly the we can see that as a second law is being governed over in the life, or metabolic process. But then even though there is disorderness, at a time you can see very orderly structure. But that doesn't mean that even though you see the orderly structures, but then you have to remember that disorderedness is way higher than the orderliness as such. And because of that, the overall entropy of the universe is going to get increased. And these are, two pictures. In first, you can see cell of the sea ocean. And you can see it's a lot of order to it over here. And this is a succulent plant. Just look into the how this plant has the leaves, and the leaves are in some kind of order here. But again, the order, basically the disorder is way higher. So when you think about the evolution of the orderly systems, is it a paradox or what? Then again, you have to think about the overall thermodynamic processes, and when you consider that thermodynamic processes, entropy or disorderness of the universe is increasing all the time. And even though life forms have evolved naturally, there is a lot of disorder, and that basically entirely that is where we have to think that the evolution, even though there is evolution, still the overall and the evolution brings the orderness in the life, but then overall there is a lot of disorderness. Again, here I'm showing one example of formation, a particular order structure inside the cell. This is the formation of lipid wall, and you can see this is hydrophobic hydrophilic head, and you have the hydrophilic tail. So this is the membrane. You can say phospholipid membrane also. And within the membrane, the hydrophilic tails are coming together because, hydrophobic tails, they are exactly opposite to each other. And the hydrophilic head side is basically exposed to the water. And there is a particular order to it. The point is that the thermodynamics dictates, it can dictate how cell works, and one can view life as such of the consequence of the laws of thermodynamics. Now I'm going to move to the next objective, that is concept of the energy currency. Again, before learning this energy currency, I'm going to give you some analogy. So before the currency as such, right now you have the rupees, dollars, euros, pounds, all the systems are there as a currency. But before that, there was a barter system, and in this barter system, there is to be simply exchange. In this exchange, let's say this person is exchanging fish with some carpet on it. This person may be exchanging a bull with the cow. So all this, it was basically way of transaction. And the way of transaction in this particular barter system, the valuation may not be, the person who is purchasing and person who is selling, it may not have a same value. It may have drastically different values itself, and that's where we have to normalize. And in terms of the having those normalization, the money concept came, the currency concept came, and then most of the time you buy the goods, you get the service in terms of the when you exchange currency, and against that you are basically getting services and all those things. Now when you think about this currency, you think just like as a salary, or within a month the person has X amount of salary, and that salary is being consumed over different expenses, and this is kind of distribution. Similarly, inside sale, this currency, which is nothing but ATP molecules also, that can be consumed for different activities. So again, when you think about the organism and its metabolic process that has energy source, and this energy source, it could be serve as a nutrient also, and from that energy source one can get useful energy. So again, this is some kind of pictorial diagram to understand the process. So let's say you have either carbon source or energy. So this carbon source or energy that can be utilized as a energy currency. The breaking down of the carbon source, that would have partial or some kind of incomplete. It could, at a time, it could be partial oxidation, or it could be a complete oxidation. And during that process, you have got some energy points or energy currencies. And these energy currencies, it can be utilized towards different objectives. It can be used for performing various biosynthesis. various repair mechanism inside the cell, then the division, excretion. So many processes are over there, and also whatever the excess amount of the energy is there, that can be stored as a that can be stored. Just like let's say you want to basically save your part of the salary, and that part of salary you basically try to utilize when it is needed, and as such for the regular use you use most of the salary, but then some part you want to store. So again, in terms of the cellular context, the energy currency is ATP molecule, and the full form of ATP molecule is adenosine triphosphate. Over here, this is the structure of the adenosine triphosphate. So, so this is adenine. It's a class of purine. You might be knowing this ending from the organic chemistry, and so I think in 12th standard also you must have learned about all those things. So when these purines or pyrimidines they are attached to the ribose molecule, then that is basically called as nucleotide, and alone purines and pyrimidines alone, that is called as nucleotide. And when nucleotide is attached with this phosphate group, then it can be monophosphate or diphosphate or triphosphate, depending upon how many phosphate groups are involved over here. In the adenosine triphosphate, what is that? You have the adenine molecule which is attached to this ribose sugar base, and then along with that you can see there are 3 phosphate groups over here, and that is how this ATP or adenosine triphosphate looks like. And in particular, in order to gain the energy from the ATP, what happens is this ATP is hydrolyzed, and this hydrolysis generates the energy. So you have this the triphosphate group over here, and this triphosphate group, if you look, it is extremely unstable because it has a negative charge, and due to that it can release the energy. So energy coming from the chemical chain to state of lower free energy. So yeah, so basically the idea over here is ATP is being hydrolyzed, and with that hydrolysis one gets the energy. Now I'm going to talk about the next objective, that is standard and cellular conditions. Now let's just when you think about this metabolism, you think about the hydrolysis or think about this this whole process. How do we basically account for it, and from the thermodynamics point of view, how do we understand it? Now, first of all, let's just talk about very standard conditions. And you take any reaction, for that particular reaction, there would be change in the energy, that is, let's say, given by delta G. And for that particular energy, there will be constant K for the equilibrium. For this, simplistic reaction, we just take about, we think about the water molecule, and let's just say that, we have temperature 290 Kelvin, 298 Kelvin, and the pressure is 1 atmosphere. And at that particular point, the concentration of H2O or water molecule will be 55.5 mole, and the hydrogen ion concentration would be 10 to the minus 7. then the change, free energy change over here in this condition can be given by this particular equation. Delta G is equal to minus RT log K equilibrium. Now, these are again, very standard conditions. And when you think about that, when you have the equilibrium constant as 1, then delta G will be 0. And just put 1 over here, and then you can see delta G will go to 0. And when K equilibrium value, if it is greater than 1, then delta G value will be less than 0. And when K equilibrium value that is less than 1, then delta G value will be greater than 0. So now, in terms of the metabolic context, we want to understand two terminologies over here. The first is exogenic reaction, and the other is endogenic reaction. But these are very different from exothermic and endothermic. Okay, these are very different. But then in the exogenic reaction, overall what happens is in this particular process, this reaction is spontaneous, and during this process the energy is released. So let's just say a reactant has this much free energy, and when the reactant is being converted into product, you can see the product free energy is less than the reactant, and whatever the difference of this energy, that is basically amounted towards the change in the free energy of the reaction, and that energy can be utilized for some other purposes. So this is exogenic reaction. In contrast to that, we have the endogenic reaction, and in this particular endogenic reaction, first of all, you want to notice that it's non-spontaneous, and the reactant has the less energy than the product. So over here, in order to make the product, it has to consume the energy, and whatever the energy is being consumed, that is basically it is basically raised with this product. So these are again the terms are very similar to exothermic and endothermic, but then they are little different. So again, exogenic reaction is nothing but in this process, in this particular reaction, you release the energy and it's spontaneous. And the other one, endogenic reaction, in this process, energy is required in order to make the product, and it is non-spontaneous. Now, coming back to the ATP molecule and its hydrolysis. So, when you think about the ATP hydrolysis, chemical reaction can be written like this: ATP plus H2O, that is converted into ADP plus Pi. And for this particular reaction, the delta G, that is a change in the free energy, that is minus 32 kJ per mole. It's roughly in that order, and in the textbook you might be see that this number is 30.5, but roughly it is the same order. What you want to notice over here, in this particular reaction, the delta G is less than 0 for the ATP hydrolysis. That implies that the change in the delta G over here should be greater than 0 for ATP synthesis. This is hydrolysis, and then for the synthesis, it should be opposite to that. That's your intuition. But then this is very interesting question. How can cell synthesize ATP if synthesis is endogenic? So again, endogenic is basically going to require a lot of energy. And over here, it's kind of again, little paradox. Paradox is ATP is used for the used by the system to have the different processes going inside the cell. But then, in order to, how does basically ATP synthesis happen? And and that is it being then what should be the situation inside the cell. So whatever we learn over here until this point, particularly this equation or so, that is basically in standard condition, and you cannot apply this standard condition inside the cell. Particularly, you cannot have 1 molar nitro concentration, which is not at all realistic. And you have to think about delta G change in the free energy, this particular context in terms of the cellular condition. And the cellular condition can be way different than the standard condition. So over here, you have to apply the mass action ratio and determine the cellular concentration also. Then what you want to modify your equation. Delta G is equal to delta G naught plus RT log Q, and that Q is basically the ratio of the concentration between product and reaction. And with that, I can basically write this equation: delta G is equal to minus RT 2.303 log K equivalent divided by Q. And where R is your this gas constant, Q is your temperature, and other things. Now, interesting things over here. Let's just say we take the ratio between Q and K equilibrium. Let's say Q is equal to 0.001 times of KQ. Then your change in the P energy delta G, that will be minus 17.1 kilojoule per mole. And for various this relation, you can see the delta G in few conditions, first two conditions is being whenever this ratio is less than 1. In that case, delta G is being negative, and when you have that ratio as well, at that point you have delta G as 0, and this will be called, this particular condition will be called as, you can say, equilibrium condition. And when this ratio Q divided by KQ, when it's more than 1, at that point, what you see is delta G will be positive. And the order, again, the magnitude, it depends on what is this ratio. So that's the, this approximation, delta G not 32 kilojoules per mole was made. But actually it is somewhere about minus 30.5. Okay. So if with this value, if I plot, this is my log of K equilibrium divided by Q, and the vertical axis is delta G. If I plot that, I get a straight line. Then, so basically that basically straight line over here. Now, how do I understand the change in the delta G in terms of cellular condition? So over here again, when delta G approaches 0, then what you think about this, as a example over here, you have this hydroelectric power station generator also, and the water is being basically released from one tank to the other tank. And when at the starting point, you might notice that it will generate the some kind of electricity also. And when water is reached at the same level in the both tanks, then there won't be as such generation of the electricity. So this particular condition, what you say is it is a equilibrium condition where there is no change. The reaction, basically kind of halting, or so basically there is no as much there is no change in the free energy. But in contrast to that, when we start again, look over here. So when we have the value this particular value delta G as a negative, that is being over here. You start over here, and in order to have the equilibrium condition, which is delta G equals to 0, So when delta G approaches 0, you are starting over here and you are reaching over here. So somewhere over here. So when delta G is 0. So how do again how do we basically understand this thing in cellular context? So when we think about the ATP hydrolysis, we can write this as a delta G equal to delta G naught plus RC log ADP divided by ATP. And the free energy available to cell from this hydrolysis of ATP molecule is dependent on number of molecules of ATP, ADP, and PI present. So in the cell, you have to understand how many currencies are available, and how exactly that currency is being dynamic. You have to exchange it, and during the exchange, the total amount of your currency will be different at a different point. But overall, you are basically trying to maintain some kind of a steady condition. So it is not basically, if you reach to, the point over here is, let's say, if you reach from this point to this point, which is delta G 0, then what would happen is cell is not going to survive. So in order to survive the cell, it has to basically work within this region where the delta G has to always negative, and that's what we are going to basically see in subsequent slides. So if you have to be in this kind of basically quadrant over here, then the cell has to basically continuously remove the product, or basically it has to replenish the substrate. So over here, the main point over here is, again, if you look into this particular reaction rate and all those things, over here, if I have to have the net free energy in terms of less than 0 or so, I have to maintain the value of Q, and that in order to maintain the value of Q, there would be continuously removal of product, or it has to basically replenish the substrate. So this is the situation inside the cell, and then in that case, what would happen? You have a steady state condition, and during the steady state condition, you will have net change of the energy less than 0. So over here, what is happening is you have a tap, and then you have the two basically containers or tanks. And with the tap, you are basically, this container is at higher height and the other one is at lower height. And this process is always going to, the turbine over here, this is going to always rotate unless you turn off the tab. If tab is on, then the amount of flow inside the container is maintained at particular rate. Then you can think that as a steady state condition. So again, coming back to this ATP hydrolysis, so ATP hydrolysis, it can release constant amount of energy only if we have continuously replenished, ATPs are continuously replenished, and this, the ADP-PI, this product is being continuously depleted. So what you have to do is basically you are using this energy, and again this has to be recycled, kind of recycled. So ADP and then PI, that has to be combined again to make the ATP. So that kind of process always goes on, and so basically the question is then how do we replenish ATP, and how do we deplete the ADP? So you have to basically synthesize ATP. That's main idea over here. So point over here again, if at all the ATP number of ATP molecules are not replenished, or number of ADP-PI molecules are not reconverted into ATP, then what would happen is eventually there would be an equilibrium situation inside the cell. And so if you think about the cell as a thermodynamic system, and if at all it has to survive, then it should not reach the equilibrium. So if if there is no free energy available to the cell, then the cell cannot work. So cell will want to avoid this equilibrium condition, and in order to have that, there would be constant flow of material, and it will have the defining feature of the life in this context would be metabolism, and which is this metabolism is never at the equilibrium. And there would be series of you can say catabolic pathways in a cell releases free energy in a series of reactions. So coming to this particular first point, when you think about it, the cell requires constant flow of materials, and that flow of materials is going to get provided by this membrane protein. And this membrane protein, at a time, they are very particular, very selective, and they work as a teeth, different kind of teeth with different pores also. So our last objective of the class is ATP as an energy currency. Now, when you think about the ATP as an energy currency, there could be three types of cellular work which are being powered inside the cell by ATP hydrolysis. One is mechanical, other is transport, and the third one is the chemical. And energy from the exogenic reaction of ATP hydrolysis is used to drive the endogenic reaction. Overall, what you think about these two processes, they are coupled reaction, and the coupled reaction, the output is exogenous. So the question is how these endogenic reactions are made into exogenic. So let's say we take a very simple example over here. Glutamic acid that is being reacted with ammonia, and then we have the glutamine. This particular process has makes change in the free energy as a positive. But then there is a different pathway or different process over here. In this particular pathway or particular process, there is a glutamic acid, and that is basically reacting towards it is reacting with ATP molecule, and after reaction, what happens is ATP is hydrolyzed. At the same time, the glutamine is phosphorylated, and then the next step would be bringing the ammonia. And when you bring the ammonia, what happens is the phosphorylated intermediate will get dissociated, and then this glutamic acid will be glutamine, and then you have the release of the gas. So this particular example over here, it is conversion is happening with the help of ATP. And the third one over here, when you think about all these molecules, let's say the the free energy over here for the glutamic acid delta G is 3.4, and the change in the free energy for the ATP molecule, that is 7.3 kilocalories per mole. So this is the coupled reaction, and in this particular coupled reaction, what you see is there is a net change in the free energy, which is negative. So for the free energy change for the glutamic acid or the glutamine, that is plus 3.4, and free energy change for the ATP hydrolysis, that is minus 7.3 kilocalories per mole. And the net change is minus 3.9. And that is how when the reactions are coupled, they are becoming exogenic as well. So this ATP molecules that will drive the different cellular processes. It can transport and it can transport a few things across the cell, and it can do the mechanical work. And then the ATP hydrolysis can lead the changes in the protein space and its binding ability. In this particular diagram, what you see over here is this is again in context of the the ATP, how ATP is being used inside the cell. So using the the transport protein, this is a membrane protein, you can say, and it can basically have it can have a selective pore, but then to have that selective pore activated, there would be ATP hydrolysis, and with that it can bring in or bring out the particular nutrients inside the cell, or it can basically excrete the nutrients outside the cell. The other example over here is the motor protein, and what you see over here is this is a cytoskeeleton tract or cytoskeleton also, and you have this motor protein, and the motor protein carrying vesicle, and this vesicle might have different nutrients, different proteins, but this motor protein has to move across this cytoskeleton tract, and in order to have that movement, it has to basically move, and what happens is the ATP is being just minute. Fine, I'll call you later and we'll next step. Okay. So this ATP hydrolysis that is going to use for this particular kind of mechanical work. And with that, I would like to stop the lecture here. And if you have any questions, okay, any questions? Okay, if there are no more questions, I can end the session here. And just a reminder, tomorrow you will have your first tutorial.